Welcome to Lecture 2 of our Nutrigenetics module titled Statistical Applications in the Great Florida Spitting Contest. In this lecture, I want to talk a little about the purpose and some applications of genetic research. One thing we do is search for genetic associations and then determine if those associations are statistically significant. Did these observed associations occur just by chance? Or is there more to the story? We work to identify relationships between genetic markers and traits, like what alleles are associated with having blue eyes, and investigate the inheritance patterns and associations between genes and diseases. How do we do this? Well, one way is to perform a chi-square test. This is a statistical method that is used to analyze categorical data. Categorical data is data that can be divided up into groups instead of numbers. For example, yes or no, red, blue, green, male or female. These are all types of categorical data. The chi-square test measures the difference between observed and expected frequencies if the null hypothesis were true. A null hypothesis is tested, which states there is no relationship or difference between the variables, against an alternative hypothesis, which states there is a relationship or difference. In other words, it is what we actually see happening. Is it very different from what we would expect to happen if there was no relationship? A super simple way to understand this is to imagine a candy company that claims each bag of their candy has a 50% red and 50% blue in each bag. When you open a bag of their candy, you count the total for each color and find that in reality, there is 30% red and 70% blue. A chi-square analysis would compare the difference between the expected numbers, which was the 50-50 split, and the actual numbers that you observed, which was the 30-70 split. Limitations and assumptions of a chi-square analysis include the independence assumption. This means that the observations must be independent of each other. Each individual observation in your data set must be separate and does not influence any other observation. It requires that observations are not related to each other. And in the context of the test, the value of one categorical variable should not affect the value of the other categorical variable. The sample size matters. A larger sample size increases the reliability of the results. A rule of thumb is that no categories should be less than a count of 5. Thus, the larger the sample size, the less likely any categories will be that small. And then finally, a chi-square is suitable for categorical data, but not for continuous variables. Yes or no questions have only 2 possible answers, whereas if you were using someone's height, the possible options are endless, really, and that would be what we call a continuous variable. Prior to performing a chi-square analysis, you will organize your data into a table with rows and columns. If you are asking the question, do college students prefer to eat at home over a restaurant? You could organize the data in a 2x2 table. The rows would be college student and not college student, and the columns could be yes and no, pertaining to whether they prefer to eat at home. Now let's take a closer look at the chi-square test and how it works. I have the actual formula listed here, but don't worry, you don't need to memorize that. We will use an online calculator for our work in this class. We're going to talk about the degrees of freedom because that is something you'll need to be able to report your chi-square statistic. The degrees of freedom depends on the number of categories and is found by simply taking the number of rows minus 1 and multiplying that by the number of columns minus 1. So for example, if you do a 2x2 chi-square, like our college student example, 2 rows and 2 columns, you would have 2 minus 1 times 2 minus 1. That's equal to 1 times 1, and that equals 1, which is 1 degree of freedom. To report a chi-square test result, there is a specific format that should be used, which is here at the bottom. All of the words in red are spots where you would insert number values. So you would write x2 parentheses, and then your degrees of freedom, which we found in this case was 1, comma, then you have your n, which is the sample size. N represents the total number of items in your sample. So for example, in our college student setup here, you would just add up all the numbers in the table. So 15 plus 47 plus 45 plus 11 equals 118. So our N would be 118. Next, we have the equal sign and the chi-square statistic value, followed by the p-value. These two values are provided in the online calculator that we will use. I plugged in the numbers from our table to the chi-square calculator, and you can see an example of the output from the website. It's in blue. It gives the chi-square statistic and the p-value and tells us if it is significant or not. We will use the 0.05 threshold, which is kind of the standard. Okay, now what is the chi-square calculation actually doing? Well, it is comparing the calculated chi-square value to the critical value and then providing us with a p-value that we can read to interpret significance. So if the calculated value exceeds the critical value, the result is statistically significant. A general threshold, like I just mentioned, for statistical significance is a p-value smaller than 0.05. And the smaller that p-value is, the less likely that the observation is due to chance. Now, the chi-square test and p-value can help us identify if there is a statistically significant association going on, but that's all it can tell us. So we really need to add more to the analysis so we can begin to understand what is really happening. This is where we bring in the odds ratio. The odds ratio is a measure of how an exposure affects the odds of an outcome by comparing the odds of the outcome in an exposed group to the odds of the outcome in a non-exposed group. You can see the formula here in the middle of the slide, which is AD divided by BC. You don't have to memorize this. We're going to use a calculator. And the odds ratio will tell us the direction and the effect that the association has. When you calculate an odds ratio, you will get a number that represents the odds of something occurring. These are often used in genetics to determine the odds of developing a disease, or in other words, the strength of the risk for developing that disease. It's important to understand how to interpret the odds ratio. So here you see that an odds ratio greater than 1 indicates a positive association between the exposure and the outcome, indicating an increased likelihood of the outcome among those exposed. For example, if you were looking at a SNP that is associated with obesity and the A allele has the odds ratio of 2.5, then you can see that the allele is associated with a 2.5 increase in odds for being obese. So in this case, the exposure would be the genotype, that A allele, and the outcome is obesity. On the flip side, if the odds ratio is less than 1, then we would say there is a negative association between the exposure and the outcome, and a reduced likelihood of the outcome among those exposed. So using the same obesity example, if the A allele for a certain SNP had an odds ratio of 0.75, it would mean that there is a reduced risk of developing obesity for those with the A allele compared to those with the other allele. Another way to say this is that there's a 25% reduced risk for obesity compared to those with the other allele. And then I just want to point out, keep in mind that these effects are actually very small in the big picture. You have to understand that the context is in comparison to like the other allele. It's not just in general. All right, let's walk through a chi-square and an odds ratio example. Let's ask the question, is there an association between brain cancer and cell phone usage? Here is a table representing people who do and don't have brain cancer and whether or not they use a cell phone. Notice the total in is 200 and represents how many participants are included. All right, now I'm going to use a website called Social Science Statistics to calculate the chi-square. The result I got was a chi-square statistic of 6.081 and a p-value of 0.03921, which is less than 0.05, so it would be considered statistically significant. When you use this website, you'll also get a Yates adjusted chi-square, but we're not using that one in this class. So what we can say at this point is that there is an association between cell phone usage and brain cancer, but that's all we can say. And since we have a significant p-value, we can go ahead and find the odds ratio, which will tell us more. Also, I've included the proper way to report the chi-square, so you can see that. It's in red and it's underlined. So using the MedCalc website to find the odds ratio, you can see this set up on the slide. There's a certain way that you put it in there, and so you can see that I've inserted the numbers that correlate with the letters that are, the order of the letters. And when I put these numbers into the calculator, we get an odds ratio of 3.05. When we interpret this, we look at the first row and first column. Those are the yes for brain cancer and yes for cell phone group. So the odds of having brain cancer is about 3 times more likely if you use a cell phone compared to those who don't use a cell phone. Pay close attention to how we interpret the odds ratio because you'll need to be able to do this. All right, now that you have a basic understanding of some of the statistics being used in genomic research, let's take a look at a study that was done right here at UF. The study is called the Great Florida Spitting Contest, or GFSC for short, and included students in the microbiology and cell science department at the University of Florida. The study began in 2018 and had the main purpose of studying the genetic drivers of the oral microbiome. Participants had to provide informed consent, a saliva sample, which was used to extract DNA and saliva microbiome information. And then they filled out a variety of surveys about diet, mental health, and other things. Genotyping was done using the PMRA from Thermo Fisher, which can identify more than 900,000 variants and other genetic markers. The diet survey is called the KidMed Diet Questionnaire and consists of 16 diet questions. There are 4 questions that would be considered negative or unhealthy, and 12 that are considered positive or healthy. If a person answers yes, they are either scored a 1 or a -1 for the poor diet choices, and the total score represents the level of adherence to the Mediterranean style of eating, which is generally recognized as a healthy diet. One of the mental health surveys is the PHQ-9 or Patient Health Questionnaire. This is a screening tool used to indicate the level of depression and what symptoms a person might be experiencing. The PHQ-9 is quite accurate in predicting an actual diagnosis of major depressive disorder by a doctor. It is a list of 9 questions that pertain to how that person has felt in the last 2 weeks. For example, question 5. Have you had a poor appetite or been overeating? The participant would indicate not at all, several days, more than half the days, or nearly every day. Other questions include things like trouble concentrating, feeling tired, or having little energy, etc. Then the scores are added up, and there are score ranges that indicate whether that person is likely experiencing minimal depression all the way up to severe depression. And one of the cutoff points that I want to make note of is if you have a PHQ-9 score of 10 or higher, that would be kind of a break off point of like being considered having depression. And we will talk about that again at a later point. So with the data collected, we can perform association studies within this student population. There are a variety of things we can look at, like gene associations with depression, gene associations with diet, etc. In our next lecture, I will be sharing my own research with you using data collected from the Great Florida Spitting Contest so you can see how this research is being conducted in real life. And that's the end of this lecture.